Yeah, what I want to talk about are these bacteriocins. And these bacteriocins are things, when you think about it, most bacteria on Earth live in very complex communities. They don't live in test tubes, which is where we grow them in the lab. They don't live in fermenters. They live in complex communities. And they have to talk to each other, kill each other, compete with each other. And one of the main ways in which bacteria communicate, and communicate right up to the level of killing each other, is by producing these bacteriocins. And if you look at the genetic makeup of most bacteria you find, for example, in the human gut or in soil, you find the genetic information to produce bacteriocins, even if you can't reproduce that in the lab and get them to produce them easily in the lab. So bacteriocins are a big part of microbial life, at least. Now, when I saw the topic of this workshop first, I thought decontamination. Well, decontamination for me is this kind of an image, you know, wiping everything out, absolutely clearing some area of all life forms within it. And that's not what bacteriocins do. Bacteriocins are not designed by nature, and they can't really be used to simply decontaminate things. And to continue this kind of military analogy, this is much more like what bacteriocins do. They're targeted. You can direct a bacteriocin against a very selective target. You can kill one bacteria in the midst of other bacteria, which is quite a neat trick if you want to make a fermented food, for example, where you want the good bacteria to grow, but you don't want the bad bacteria to survive. Or many foods where bacteria contribute to shelf life and stability and so on. You don't want to kill everything, you only want to kill the target bacteria. And bacteriocins actually come in both kind of modes. Some bacteria are incredibly selective. They only kill a very narrow spectrum of target bacteria. Some are quite broad and kill, almost do decontaminate the environment in which they live. So bacteriocins are a tool that bacteria use that we can potentially exploit. And we've been doing this for about 15 or 20 years now, together with Paul Ross in Moore Park and myself. We've what we call the Cork Bacteriocin Group. We've spread across the two institutions. Generally, there's about 15 to 20 people in that group, about half in UCC, half in Teagasc, and then we have other areas of interest as well, which partially overlap. But bacteriocins are really interesting. If you look at this, if you can see this image here, what you're seeing is bacterial colonies growing on a plate, and then the plate has been overlaid with another bacteria. And you can see that these producer bacteria, the colonies, have produced something that are killing the target bacteria. But there's a lot more information actually in this image. If you look at it, these bacteria here are killing these bacteria at, when microbial terms, is an enormous distance. If you blew a bacteria here up to human size, that's 20 kilometers. That's a 40-kilometer diameter. That's me standing here killing everything within 40 kilometers diameter of this spot. So these are massively active. They're active in about 1,000 times more active than the best antibiotics. So these aren't antibiotics, these are incredibly active. The other thing you can see is that it's being secreted by living cells. It's not as a result of cell disruption. And there's quite a lot of it being produced relative to their activity. There's a double-edged sword. They're active in such low amounts. They're active actually in the single nanomolar range, that therefore the bacteria only have to produce very little. So very often when we deal with food companies, they say, we're interested in your bacteriocin, send us 100 grams of it and we'll see what we can do. We say 100 micrograms would be a big deal for us. But what is it you want these bacteriocins to do? Because it might be that the 100 micrograms can do it. The other thing you can see from this picture is that the bacteria producing the bacteriocin are obviously immune to its own action. Otherwise, it would be a suicidal thing to do. And so the producers are immune. And most bacteriocins that have been characterized are active against gram-positives and produced by gram-positives, but there are gram-negative bacteriocins as well. And then they come in various different classes, which is of interest to us who work in bacteriocins, but probably of less interest to a more general audience. And in terms of mode of action, a lot of work has been done in this area. We've done some of it, but some of the best work has been done in other countries as well. In Germany and France, they've done some beautiful work on this, in Holland. And these papers describing these mode of actions are published in Science and Nature. I mean, this is really fundamental molecular biology. And what they've shown is that there are various different classes. What they all do, or what the main class of bacteriocins 1 and 2 do, is they get through the cell wall and they attack the cell membrane. They form pores in the cell membrane, much in the way the pulse electric fields do. These are quite stable pores, And once you form a pore in the membrane, there's no differentiation between inside and out, and therefore the cell essentially dies. And this happens within nanoseconds of the action of the bacteriocin. And mathematically it's been calculated, although no one can understand why this should be so, because you need usually about 4 to 6 molecules of the bacteriocin to form a single pore. But mathematically it's been calculated, when you look at those diameters and so on, that maybe 1 to 2 molecules of a bacteriocin can kill a cell. And I sometimes talk to undergrads, use the analogy of that single torpedo down in the right part of the Death Star in the Star Wars. Just one bomb in the right place kills the entire Death Star. One bacteriocin in the right place, one bacteriocin molecule can maybe kill a cell. They have other modes of action as well, but I think, again, that's beyond the scope of this discussion. The other point I wanted to make, and if you're a molecular biologist, you love slides like this. If you're not, you'll absolutely hate them. But what I'm showing is the production of niacin, one of the most industrially used bacteriocins. And there's niacin there outside the cell, but to get that there, you need 11 genes. You need a very complex production machinery, introducing all these post-translation modifications, getting it outside the cell, protecting the cell against its own activity. There are sensor mechanisms. As I said, bacteriocins can be used as signaling between cells. So it's not trivial to produce niacin. It's a very big investment by the cell in terms of the DNA, the biochemistry, the energetics to get these things out of the cell. This is not a trivial thing to do. So when they produce it, it must be because they really need to produce it and want to produce it. So niacin is one of the most industrially used bacteriocins. Lactocin 3147 is the Cork bacteriocin. We discovered this in Cork back now in 1995, so a very long time ago. And over the intervening years, we've solved the structure of it, which wasn't, again, an easy problem. We did that together with John Vedras, a chemist in Edmonton in Canada. We've solved the NMR structure. We know how it works. We know the mode of action. It's a very interesting bacteriocin. Again, not the time or place to go into it, but it has some very unusual characteristics. And we isolated this from a strain, from a bacteria donated to us by a farmer house, someone who used this particular bacterium that makes this to make buttermilk, to make bread. And so it's unique to Ireland. We're the only ones that have it, and it's patented and licensed and so on by UCC. And lactocin is our kind of flagship bacteriocin. We try and develop applications for lactocin. We have about 20 or 30 other bacteriocins in the arsenal that we work with as well. And just to give you a sense of the potential value of lactocin, here's lactocin against Staph aureus. Now don't worry about the zone size so much here. These are just different antibiotics against Staph aureus, and there's lactocin. Obviously, if you use more lactocin, you get a bigger zone. If you use less penicillin, you get a smaller zone, and so on. So don't worry too much about the zone size. We see that Staph aureus is sensitive to all these antibiotics and to lactocin. But if you take the much vaunted MRSA, the multi-resistant or methicillin-resistant staph, that one is resistant to all these other antibiotics, still somewhat sensitive to vancomycin, which is the last line of protection against MRSAs, but lactocin continues to be active because it actually attacks through a different mechanism. There are now vancomycin-insensitive staphs as well, and lactocin still kills them. So there's a potential for using it in places where antibiotic resistance is growing. Bacteriocins don't use the same target sites, and therefore they're impervious to whether something's drug resistant or not. And what we've been interested in doing is developing applications for these bacteriocins, and we've ranged across food quality applications, food safety applications, veterinary medicine, human medicine. And what I'm going to do in the next 10 minutes is just touch down on some of those examples, just give you a sense of how useful these bacteriocins can be. It's a very cheap technology as well, I should point out. But various different applications that I'll maybe touch on again. I don't think I'm going to come back to this one, so I'll just say it now. We use lactocin-producing strains to protect against mastitis in cows, and we've used it very successfully. But I could give people details on that if they're more interested. And I think I'll be touching on some of the others in the next couple of slides. So one useful thing about having a bacteria which produces a bacterium, and I should have mentioned already that the bacteria that makes lactocin is Lactococcus lactis. And Lactococcus lactis is the cheese-making bacteria, completely safe. We eat it and it's billions, trillions anytime we eat cheese. So it's permitted for use in foods and so on. It's a grass organism, generally regarded as safe. And what we can do is we can either put the live culture directly into a food which already contains live cultures, like cheese itself, or we can develop an ingredient based on the bacterium and then put the ingredient into either fermented foods or non-fermented foods. And as I said here, it's food grade, it's economical. This bacteria will grow in milk, and then you simply dry the milk back down to a powder. Bacteriocins are completely heat stable. They can withstand boiling for 20 minutes, 30 minutes, certainly withstand pasteurization and spray drying. You need to autoclave them really to even make a dent in their activity. So then you can incorporate this dried powdered ingredient instead of maybe skim milk powder in different foods. So live cultures is one thing you can do. Here's an application that we developed very early on. I shouldn't have had so many bills on this, but basically what we can do is we can take the original strain isolated from this bacteria that makes buttermilk for making bread that we got from a farm in the middle of the country, and that produces lactocin. And one thing that turned out to be very useful very early on was lactocin also needs a lot of genes to make it, 10 genes, you can see here, but they're on a plasmid, and that plasmid can be moved from strain to strain. So we can take commercial starter cultures that are used in cheese making, in making fermented meats, fermented fish, fermented vegetables, and we can transfer the plasmid across very easily. And now the new strain will start producing the bacteriocin. And the important thing to notice is these are not genetically modified organisms. These are natural. You're allowed to use them straight away. You don't need any regulatory permission. So if someone's making cheese with a particular strain, we can transform their strain to become one which produces lactocin, and they just carry on making their own cheese again, but now it produces lactocin. Here you see the plasmid from the original strain. There's the plasmid content of the commercial starter strain, and there it is now having acquired the new plasmid. So this is very useful because it means they're all food grade and we don't have to hide them in the lab. And this application, which again I'll just bring up in its full glory, shows us that generally when you make cheese, for example, this is a very important application for the cheddar cheese industry, particularly in Ireland, which is a big industry for us. And what you see is when you make cheddar cheese, the starter bacteria, the Lactococcus lactis that you make the cheese with, that acidifies the milk, that dies off over the ripening time. We're looking here at, say, out to 6 months. But it's replaced by something called a non-starter lactic acid bacteria population. And when you look by pulse or by genetic fingerprinting, you see that's a mixed population of bacteria. They come in from the cheese factory. And they're what gives cheese its characteristic flavor. A decent cheese taster will be able to tell you, even by rubbing the cheese and smelling it, we'll be able to say, that's from Kerry, that's from Glanbia, that's from Dairygold. And certainly what they do is they just rub a piece of cheese, lick their thumb, and then they can get the flavors and the volatiles and so on. It's called cheesemaker's thumb. And so these are very important for giving flavor, but they can also give defects. They can give gas, they can cause late gas blowing, they can cause off flavors. So cheesemakers really would like to control this, but they can't with current technology. If we make a cheese with the lactocin producer, so we just swap that strain for one that produces the bacteriocin, we now get essentially a cheese which is sterile except for the producer itself, which still dies off at the same rate, but there are no bacteria in it. And even if you melt that cheese after 9 months and put it into an agar plate, you can see that the cheese itself now is full of this bacteriocin. Now what we can then do is we can take a nest lab that we like, a probiotic nest lab or a nest lab that gives a very beneficial flavor, and we can make that slightly resistant to lactocin and superimpose that on the background, and now that will grow essentially in this background, and we get a cheese which has only one bacterium in there, the one that we want, the one with the proper flavor benefit or the probiotic benefit or whatever it was. So you can see this is a zero cost game. All you're doing is you're swapping one starter for another starter. There is no technology, there's no investment, there's nothing. This is zero sum. And you can completely take over control of what your cheese is going to taste like, look like, smell like, and so on. So this is one particular application which is a very useful one. We can obviously extend that into things like controlling pathogens. Here's cottage cheese, for example, made with the normal starter. and then spiked with Listeria. Now what you're looking at in these circles are not the original starter. You're looking at the Listeria monocytogenes, and Listeria survives very well in cottage cheese. It's not very low pH. But in the cottage cheese made with the lactocin-producing starter, it's eliminated. Same here in salami, Staph aureus in this case we're killing. And here's Listeria monocytogenes on a mold-ripened cheese. But again, zero cost. So you're making the cheese at the exact same cost. There's no change. Just swap out your starter for a starter producing lactocin, and you get control over all these pathogens at zero cost. Now, moving into the area of cost, or if you're not making fermented foods, but you're making some other kind of food, what we do is we simply take milk our whey, we essentially sterilize it or UHT treat it, we add the bacterium, we ferment it, and we ferment either the milk or the whey. We dry it back down again. Now, high temperatures involved in that, but that's no problem for the bacteriocin. It survives those temperatures. And so you end up with a powder, either a milk powder or a whey powder, which is enriched with the bacteriocin. And then you can add this as an ingredient to food or resuspend it or do whatever you want with this, where you'd normally use normal milk powder or whey powder. Now, of course, there's an expense involved in producing this, but nonetheless, it's very cheap and it's using existing plant machinery. And now if, for example, you put this powder in place of the normal whey powder in baby formula, for example, and you spike that baby formula with Listeria, Listeria grows in normal baby formula. In the baby formula, where about a third of the whey powder has been swapped out for the lactocin whey powder, you get complete control. So again, there is a cost involved here, but it is quite effective. You can do the same thing in things like yogurt. You can just mix in the whey powder or the milk powder into natural yogurt, into rehydrated soup. There's an example there with a spore former, Bacillus cereus, and adding in 1% or 5% of this whey powder into that particular soup. So very economic and useful technologies. This one I like a lot. Well, we've never got any traction on it in Ireland because I think milk manufacturers are quite happy that after a week or so the pasteurized milk goes off in the fridge and someone has to go out and buy more. But in other countries that's a big problem, where pasteurized milk has a very limited shelf life. And what we've done here is at 4 degrees and at 8 degrees, we've simply mixed some of the milk powder back into the liquid milk before pasteurization. Then we go ahead with the normal pasteurization. And whereas normally pasteurized milk gives like a microbial bloom after about 2 weeks at 4 degrees, and after about a week at 8 degrees, if you add more and more bacteriocin, you get essentially a month shelf life. Even at 8 degrees, we'll get a month's shelf life for pasteurized milk, where the microbial bloom never happens. And we have worked out the cost of this at about 0.5 cent per litre. So it's not expensive technology, it's very cheap. We can also incorporate...
And those ones, are they all in production in different places?
No.
Or no, these are...
No.
Yeah.
Still in the lab. We can incorporate lactocin into food packaging. This is work we did with Elke Arendt in UCC, who's a food technologist. And we can incorporate lactocin or niacin into various different membranes, inserts between, for example, go in between cheese slices or in between meat slices. And then we can see good control again over things like staph and listeria in meat and in dairy products. So it can be incorporated into these kind of things, and it can even be bound onto solid surfaces as well. There's also synergy between lactocin and some of the other if you like, high pressure. We haven't tried pulsed electric field, which is something that would be worth trying. But here, for example, you get the combination effect, synergy, well beyond additive between lactocin and high pressure. And if you put a bacterial cell under pressure, you seem to make it easier for the bacteriocin to get into the membrane and form the pores. And so there's a good combinatorial effect. So if you want to combine it with high pressure, and probably with some of the other technologies as well, it'll have a good outcome. And then we've been playing some games. We try and interest companies in these kind of things. But here's one, for example. It's very active against Streptococcus mutans, which is the anchor bacteria for plaque, the anchor bacteria for causing plaque and tooth decay. And if you look here, you can see that in saliva, this is done in both in artificial saliva and in volunteer collected saliva. You can see that if you add lactocin to Streptococcus mutans, you get an initial 5 log kill almost within minutes. And at lower concentrations, it takes a bit longer, but you still get a very good kill. And that again, you can detect the lactocin from cheese, from yogurt, from anything that's made with the lactocin producer. And we can even get up to about 100,000 activity units per mil in some of these powders that we make. And one of the things we're trying to do, trying to interest one of the big Irish companies in, is making a cheese which is impregnated, if you like, with the lactocin. A particular cheese, I won't mention the proprietary name of it, which comes in strings and can be pulled apart and is very tough. And you get a kid to chew on that, it would kill the strep mutans in their mouth, and it'd be something they'd maybe put in a snack box for after their chocolate snacks or whatever. They could have some beneficial impact. We've done some tests in rats, but not in humans yet. Let me turn back for a second to niacin, because some people in the food industry will have some experience with niacin. It's E234 is the additive number in Europe. Here you see it on a particular can of tapioca in this case. The preservative is niacin. It's also one of these very complicated bacteriocins. It's used in all these kind of products, and it's actually a big product. It's got a big market. Does Kerry make it in Ireland? Well, they make it in the US, but they make Kerry Group make it. Danisco make it, their Chinese companies make it. It's a very big commercial ingredient that's used around the world. And it's used in a lot of different foods to take advantage of its heat stability and its very high degree of activity. There is one target which is our favorite bacteria against which it's not very good. And so here's the niacin is actually one of the commercial names of the commercial forms of niacin. Here it is controlling listeria in ham paste. And what you can see is that, unfortunately, it doesn't offer very much control. Niacin doesn't really work. Another bacteriocin, pediocin, works very well and managed to restrict growth of listeria in ham paste very well. So sometimes you have to do horses for courses. If you chose niacin in your ham paste to protect against listeria, you're in big trouble because it's just simply not going to work. A different bacteriocin does work. So you've got to pick and choose. That's why I'm saying it's not just a decontamination strategy. You've got to pick wisely. But we set out to kind of solve this problem. Why is niacin not very good against listeria, and could we do something about it? Can we bioengineer niacin to improve its efficacy? So first of all, what we did in the lab was we tested, is it any good against listeria? And there's listeria classic growth curve, and there it is in the presence of niacin, the kind of amount of niacin you'd use commercially. And yeah, you might get a slight lag, but it really is not very effective against niacin. And so what we want to do is take advantage of the fact that it's a genetic basis to the production of niacin. There's a gene for niacin. Even though it takes 10 other genes to produce it, there's still a gene which encodes niacin. So what we do is we go in and we do random mutagenesis of that gene. We have a robot which allows us to screen about 2,000 to 3,000 individual colonies per day. We can screen them by overlaying them with the target organisms we're interested in. There's the structure in niacin. After about 3 months of this, where the student was really at the end of their tether, because I'd screened now at this stage something like 200,000 niacin variants, we had that wonderful eureka moment that you get sometimes in the lab, where this thing emerged, where this is the normal wild type producing niacin, and this thing came out which is producing far more niacin. Either far more niacin or a much better niacin. They were the two options. So we went back and we tested that. We found that the mutation that I showed you was in that amino acid there, in that lysine. So what we did was we went in now and did a very direct mutagenesis. We changed every one of these 3 amino acids to each of its 19 possible alternatives. And then we did all the various combinations. We generated in all something like 24,000. But in this case, we knew exactly which change we'd made at every single position. And we tested all those. And that gave us a bigger bank. We now have ones that we call things like N20P, which means the asparagine at position 20 is changed to a proline. And that one is more active than the wild type. We've got M21V, which is that one changed to a valine. And we got K22T, K to T, or K to serine. That was the original one that came out, and it came out again. So we were very happy about that one re-emerging. And just to convince the skeptics, here's the amount produced of each of those by the bacteria. So it's not that it's producing more, it's producing a more active one. And when we went back and tested that, we saw that There again, original growth of Listeria. There's the growth of Listeria in the presence of ordinary niacin, and there it is in the presence of our new niacin equivalents. And we've just signed a license with the main niacin producer, the world largest producer of niacin, to start producing niacin V, which is our new variant. We're in the very early stages of production now. UCC will take all this, I won't get. And here, for example, we've done the same trick. Once you get a trick that works, you just keep on doing it over and over again. So we start asking what are the other problem bugs that niacin isn't very good at. One of the others was MAP, obviously a dairy-related issue. And here's the wild type. And here we see various different versions which are much more active against Mycobacterium. Smegmatis as a control here, but we've now retested them against MAP, and they're also much more active against MAP. So we're playing around with other targets. So if somebody is aware of a target and you want to get a new niacin for it, we'd be willing to screen. The other thing that we use bacteriocins for are as probiotic traits, because again, remember, if this is a probiotic organism and this is a pathogen that causes disease, you're in a very good area. And so what we're showing here is a mouse experiment. We engineered the Listeria to emit light, and we can then follow the infection in real time. So these mice are alive. They can be looked at all through the day. They can be looked at hundreds of times without sacrificing them. So we get lots of time points. You can see this mouse is infected with Listeria. And Listeria goes to the liver and the spleen, causes a massive infection. This mouse received a probiotic. The probiotic produces lactocin. This mouse here got the exact same probiotic with the single gene knockout, so it no longer produces lactocin. And so you can see that the entire effect is due to the production of bacteriocins. And this is the only instance so far, I think, where a probiotic effect has been brought down to one single gene within a particular probiotic. But having seen that, we thought, this would be fantastic if we could do this for some of the real problem hospital bugs. So we've targeted Clostridium difficile. Here's C. diff. The problem with C. diff is a disease caused by antibiotics. You go on antibiotic treatment, kills all the bacteria in the gut, or many of them. The C. diff are spore formers. They survive, they grow, they produce toxin, they kill you. You try and treat it with antibiotics, which kills the background bacteria, so you get recurrence and cycling. We looked for a bacteriocin. We tried to take advantage of the narrow spectrum to say, let's find a bacteriocin that only kills C. diff and doesn't kill anything else. Now, again, that was a bit of a wish, but Mary Ray, as part of her PhD, screened over 50,000 spore formers from the human gut. And one day, again, she had her eureka moment. There's a bacteria producing something which is killing the C. diff at a huge distance, but not killing any of the other bacteria that were isolated from the gut on the same plate. So the bacteriocin was going past all these other gut bacteria to kill the Clostridium difficile. And we, again, sequenced the genes, and we identified the bacteriocin, and we solved the structure, again, with John Vederis in Edmonton. And it's, again, a very novel kind of bacteriocin. This is not the time or the place to go into what it does. But what's very interesting about it is it's very active against C. diff. You see here, killing C. diff. No activity whatsoever against lactobacilli, bifidobacteria, the kind of bacteria you find in the gut. So they ignore it. This one is rapidly killed by it. And we then tested it not in humans, because we can't do C. diff experiments in humans, obviously, and there isn't any really good animal model. So we tested it in a mock-up of the human gut. This was developed by Glenn Gibson in the University of Reading, and it's essentially a chemical replica of the human gut. And we compared Turacin, which is our new bacteriocin, against vancomycin and metronidazole, which are the frontline drugs for the treatment of C. diff. And we looked not only at the fate of the C. diff, but at the fate of all the other bacteria present in the gut. And we did that by deep sequencing, by pyrosequencing. And what you can see is that in the normal situation, don't worry about the fact there are two controls here. In the normal situation, the control, the C. diff, which was inoculated at this level, grows about 10-fold over 24 hours. If you use vancomycin or metronidazole, it's killed, as you'd expect. Turacin is just as effective as vancomycin or metronidazole, the frontline drugs, at the same concentration at killing C. diff. So that's the kind of headline, if you like. What's more interesting is what's going on behind that, what's happening to the rest of the bacteria in the gut. And we can look at that by deep sequencing and kind of draw a pie chart. This is at the level of phyla, and it's going to be very difficult for you to see, I think, but there's a pie chart with different colors here, and that represents the normal distribution of phyla in the human gut. When you add either thoracin or when you add either vancomycin or metronidazole, you completely destroy that normal distribution. You see these green ones, if you can see it, have completely dominated, and they're proteobacteria, things like E. coli and Salmonella and Shigella and a lot of bad bacteria in that lot. While the one that was treated with thoracin is unchanged. So you can kill the C. diff without killing the background flora, and that's the benefit of having something which is very narrow spectrum. This again has been licensed to a small Irish biotech company which is now seeking major partners. But what it looks like is a kind of a magic bullet for C. diff. Again, I don't know if you can see it, but here's a zone of killing against C. diff, and here are 5 of the most predominant industrially used probiotics. This is the Nestle one, this is the General Mills one, this is the Actimel and so on, this is the Yakult one, and you can see that it has absolutely no effect against any of those. So we're going to try and come up with a kind of a treatment that when you go into hospital, if you've got a risk of C. diff, you go on probiotics and thoracin rather than going on these very heavy broad spectrum antibiotics. So in conclusion, bacteriocins are antimicrobial peptides, can be used as tools to manipulate the composition of complex microbial flora in food and in vivo. It's not really about wiping everything out. It's about going in and shifting the balance or getting rid of something that you don't want to be there while not affecting everything else. But you have to be careful. And I was really curious. I was going through, we were dumping some stuff out of the old food science library, and I came across this, and by pure chance picked up this one issue. It was August 1958, and in it they had an article on niacin in dairy products. So this is a 70-year-old technology. But this resonated with me. The indiscriminate and unintelligent use is most likely to lead to disappointment and frustration. And I think that has been a big problem. People say, I have a problem, I tried niacin, it didn't work. Bacteriocins are no good. And that's the end of their exploration. You've really got to, I would just expand that to all bacteriocins, but it's a horses for courses. You have to almost come up with a designer solution for your problem. Otherwise, it's really going to lead to a lot of disappointment. So if they are so effective, why aren't they being used in more foods? I think they're very confusing regulatory signals. In Denmark, for example, they say you can't use a bacteriocin producing culture in a fermented food, but you can if you don't know if it's producing a bacteriocin, which is a very difficult regulatory thing to get around. There was a Danish head of EFSA, and it was to me, and I'm definitely making a few leaps. Somebody mentioned bacteriocins as potential antibiotics, and she just set her face against, we shouldn't have any antibiotics in food, therefore there should be no bacteriocins in food, therefore we're banning bacteriocins in food in Denmark. And then when people said, but most of the bacteria we already use to make food in Denmark have bacteriocins, she went, so long as we don't know about it, we'll let it lie. So that's a funny kind of a situation. Obviously they are permitted in most countries in Europe, and I'd say if the Danish case was tested, it would be okay as well. There's a lack of appreciation. This is the point I was making on the last slide. Solutions may have to be tailored to particular problems. It's not necessarily an off-the-shelf solution. It may be something you have to kind of work through. And definitely, and I hope I haven't done it, but I probably have, is an overhyping of bacteriocins. Some people present them as if they're a solution to everything. They're not broad-spectrum antibiotics. They do not render a food safe just by simply being present. You have to tailor-make each solution. And I finish by thanking Paul. You can't see most of the people there in the group that was taken a couple of years ago, but that's John Vedras, the Canadian scientist we do most of our collaborations with. Paul is head of food research in Chagas now. The Cork Bacteriocin group, of course, are funders, some of our collaborators. Carl for inviting me, Paul for your hospitality, and the rest of you for your attention. Thank you very much. There's an odd, again, regulatory kind of grey area. If you concentrate a bacteriocin from a fermentate or from a food acceptable source, you do not have to label it, regulate it, do anything with it. If you purify it, you must now seek an E number. So what most companies who make bacteriocins do is they make fermentates. And they just say, our fermentate will give you a longer shelf life, it'll improve food safety. It's a fermentate. They'll never actually say the word bacteriocin unless you ask them, how does it do it? And they say, well, it's because it's full of bacteriocins. But if you want to purify bacteriocin, niacin is purified to a degree. It's about 2.5% pure, and then the rest is just skim milk powder. They had to have an E number. So if you want to concentrate it, just simply dehydrate, if you like, you're fine. But if you want to purify it to any degree,
to extract out
the active, then you've got to get E numbers and so on. It's really frustrating. We've dealt with a lot of companies on bacteriocins. We've taken some very far towards product, and there's always some issue. There's always someone in the company. The biggest thing that's dogged us is people taking bacteriocins away and then coming back to us saying, it didn't work. I said, what didn't work? And they say, well, we contaminated some with salmonella and it didn't work. I said, of course it didn't work. They don't kill gram-negatives. I say, yeah, but everyone's turned off it now. Kind of, we're all fed up with it and we tried it. And so we're working with companies now that know bacteriocins and if you like, shared minds trying to co-develop something. I think in the past we tried to present it almost as we have a solution, and the company took it and said, well, it didn't solve every one of our problems and we're not interested anymore. So it's...
involved a bit earlier, maybe, and they think it's part of their...
Well, anyone who's used them, Danisco are major producers, well, DuPont now, they've been bought out by DuPont. They're major producers of bacteriocins, as their Kerry group, and so they know the field, so they're much more interested in working with us, and that's the kind of companies we're working with now.